Assalamualaikum and Salam Malaysia Madani. This is Group 6. We will present about blood collection and smear preparations in fish. I hope all of you enjoy this presentation. First, I would like to introduce all of you to our team member consists of 5 persons. First, Sharul Effendi bin Samsul Bahari. Second, Muhammad Rizal bin Sokraji. Third, Norafida binti Haji Maidin, that's me. Fourth, Nosri Sharizan bin Abu Bakar. And the last one is Zulhami bin Amin. Ladies and gentlemen, in this introduction, I will explain about blood collections and smear preparations in fish are vital techniques used in research and aquaculture. They provide crucial information about fish health, disease status, stress levels, and reproductive status. These procedures aid in disease diagnosis, environmental impact assessment, and scientific studies. Proper implementations ensure the welfare of the fish and informs decision for better management.
Hello everyone, my name is Muhammad Riza. Why is blood collection important? The management of fish health must include blood sampling. Blood sample can be collected and examined to help us spot disease early on, track the success of therapy, and spot potential environmental danger. Making educated judgment about the well-being and health of our fish population is made possible with the use of this information. Blood collection does carry some hazard, so fish and collector make it hard due to improper equipment handling or untrained employer. To reduce this danger, it's crucial to apply the right safety procedure and tools. The advantage of collecting blood outweigh the possible risk, notwithstanding this difficulty.
The blood collections in fish is crucial for various research and diagnostic purposes, especially in the field of aquatic biology and veterinary medicines. The material we are list are indeed commonly used for fish blood collections and analysis. Here a brief explanation of the usage. The first is live fish. We use as the subject for blood collections. The others is anesthetic solutions, such as trichine metanosulfonate MS-222. We use to sedate or anesthetize the fish during the blood collections procedure to minimize stress and discomfort. The others is sterilized PBS or phosphate buffer saline. We use as a balanced salt solution to dilute the blood sample and maintain its isotonicity. The others is sterilized syringes, 1 ml or appropriate size. We use to draw and measure the blood sample. The other is sterilized needles, 23 to 25 gauge or appropriate size. We use to puncture the fish blood vessels and collect the blood samples. The others is anticoagulant, such as heparin, sodium citrate, potassium, ethylene, diamine tetracetic acid or EDTA. We add to the collect blood to prevent clotting and preserve the sample for analysis. The other equipment we use is microscope slides and cover slips. We use to prepare blood for microscopic examinations. The second is tubes used to collect and store blood samples with or without additives, or we can call it pipette. The hemocytometer are specialized slides with grids used for manual cells counting in the blood samples. The microscope we use to examine the blood smears and analyze blood cells morphology. The Rexten or Giemsa staining reagents applied to blood smears for better visualizations and differentiation of blood cells types. The disposal glove and lab coats. We use the personal protective equipment PPE to worn by the individuals collecting and handling the blood samples to ensure hygiene and safety. The blood the fish blood collections and analysis are essential for understanding the health, psychological response, and overall well-being, particularly in aquatic research and fish health management. The proper handling and use of these materials are crucial to ensure accurate and reliable results while minimizing stress on the fish during the procedures.
Ada sudah. Mantap. Berapa? Empat. Ini untuk sampel pertama kan? 100 ya kan? Dalam dua yang lain. Banyak sih ya. Biasanya 0,25 kan?
Itulah tuh.
Oke.
Ini boleh lah kan?
This picture shows the diagram for taking the blood samples. The blood collections in fish is an important procedure in research and diagnostic studies. The common method include the tail ablation of euthanized fish. The tail ablation is not a blood collection method, but a postmortem procedure to remove the tail fin of euthanized fish for various research purposes. The caudal fin puncture is a blood collection from small laboratory animals such as fish by inserting a needle into the caudal vein located on the underside of the tail. The third is cardiac puncture, the blood collection from fish by inserting a needle or cannula directly into the heart cavity after euthanization. The fourth is a dorsal aorta puncture, the blood collection from fish by inserting a needle or cannula into the dorsal aorta, or major blood vessels running along the fish backs, typically after anesthetizing the fish.
Hi, I'm Sharul. Welcome to my slide. I will explain about the blood smear and staining procedure. So, a blood smear is a common laboratory technique used to examine blood cells under the microscope. It is the essential part of the complete blood count (CBC) and can provide valuable information about the different type of blood cell, their morphology, and any abnormalities present. Staining is the performed to enhance the visibility of the cell and improve the contrast under the microscope. Here the step-by-step guide on how to prepare a blood smear and perform the staining. First thing first, prepare the blood sample. Before making the blood smear, the blood sample need to be adequately mixed to ensure the even distribution of the blood cell. Gently invert the tube, contain the blood several times to mix it properly. Second, prepare the slide. Place a clean glass microscope slide on the plate. Then we clean the microscope slide with the alcohol and allow it to dry. After that, collect the blood using the sterile disposable pipette or capillary tube. Draw a small drop of the blood from the well-mixed blood sample. The drop should be about the size of the matchstick head or less. After that, we spread the blood. Hold another clean glass microscope slide at a 30 to 45-degree angle from the first slide. Touch the edge of the second slide to the blood drop on the first slide, allowing the blood to spread along the edge of the second slide. Capillary action will help the blood spread across the width of the slide. After that, we go to the smear the blood. Quickly and smoothly push the second slide away from the drop, spreading the blood along the surface of the first slide. The smear should be thin and even to allow proper visualization of the individual blood cell. Lastly, air dry the smear. Allow the blood smear to air dry completely. This usually takes a few minutes to ensure the cell adhere well to the slide. After we do the blood smear, we go to the staining procedure. First one is fixed the smear. Second is staining, which the commonly use the staining for blood smear and the right stain and the GIMSA stain. Follow the manufacturer instruction for the specific stain are you using. The general stain procedure involve the covering the blood smear with the stain solution for a specific period. Three, washing and drying. After the staining period, rinse the slide gently with the distilled water to remove excess stain. Pat the slide dry using the blotting paper or a lint-free cloth. Lastly, we mount the slide. Place the Microsoft cover slip over the stained blood smear using a mounting medium to facilitate the placement of the cover slip. The result is using the appropriate magnification. The different blood cell type can be observation and analyzed based on their size, shape, and staining characteristic. The examination can provide valuable information about the patient health and aid the diagnosis of various blood-related disorders.
In conclusion, we have learned about the importance of blood collection and smear preparation in fish. By collecting and analyzing blood smear, we can identify potential health issues and take proactive steps to ensure the well-being of our aquatic. We discussed the different type of blood collection method and equipment, as well as the preparation and staining technique used in process. We also cover how to interpret blood smear and identify common blood disorder in fish. By following best practice for blood collection and smear preparation, we can ensure the safety and ethical treatment of these animals. Okay, that's all. Thank you from us. Bye bye.